Welcome to Lecture 11 of the Cardiovascular section of the Integrative Systems Physiology course. So today we will be talking about reflex regulation of the cardiovascular system and how reflexes influence the degree of autonomic nervous system outflow, degree and directionality of that outflow to optimize cardiovascular homeostasis. And during that last lecture, what we discussed was how the autonomic nervous system can influence cardiovascular function. Of course, the big question being then what actually influences the influencer, if you will. And as alluded to at the outset here, it is reflex regulation of autonomic nervous system outflow. And so therefore, the biggest question here is, well, what are the reflexes that are modulating sympathetic and parasympathetic activation and the consequent physiological responses that maintain homeostasis? And a major influencer of autonomic nervous system outflow to the cardiovascular system is, of course, baroreflex function. So baroreflexes are critical for a sensation of changes in systemic perfusion pressure and vascular distension, and subsequently are reflective of changes in either vascular tone or cardiac output. Therefore, it serves as a terrific modulator of autonomic nervous system outflow. Now, when we look at where these baroreflexes are located, they are located in the aortic arch as well as in the carotid sinus. So evolutionarily, of course, this makes a lot of sense because if you have baroreflexes at the site of the aortic arch, that is a terrific place. This is rated the outflow tract of the aorta, or if there are sudden reductions in cardiac output or stroke volume, then those will subsequently be reflected in reductions in distension of the aortic arch and very rapidly be sensed by the aortic baroreceptors and send a signal up to the brainstem saying, hey, we should be altering autonomic nervous system outflow to compensate for this. Now, similarly, with placement of the carotid baroreceptors, makes a tremendous amount of sense to have those at the major perfusion sites and entry sites of blood to the brain. So, of course, even if the aortic baroreceptors are functioning appropriately, or if they fail to function appropriately, then you still have a signaling mechanism here at the inflow sites to the brain to ensure that if perfusion pressure to the brain is reduced, then you end up having profound signaling inducing reflex changes in autonomic nervous system outflow that will aim to compensate for that and preserve brain blood flow. When we look at these receptors in a little bit more detail, you can see here that in the carotid sinus that there is robust innervation here at the carotid bifurcation and in the sinus. In the aortic arch, there's profound innervation that extends around substantial portions of the aorta. Very minor innervation here in the pulmonary artery as a sort of earlier sensor for reductions in right ventricular outflow. But again, really focused on the aortic arch and outflow from the left ventricle. And these baroreceptor fibers are very densely innervated in this region, which of course then is optimized for a sensation of reductions in distension of the aorta and subsequent modulation of sympathetic and parasympathetic outflow. Now, when we look at baroreceptor firing rate, it is interesting because firing rate increases with increases in stretch, or for example, in hypertension. So, like most nerves, you don't have activation of a nerve or activation of a sensory fiber and then a reduction in firing rate. So here, this is no different. So when you have activation or stretch of the baroreceptor fibers, what you have is an increase in firing rate. So then by default, that has to send an inhibitory signal to the brainstem. And that inhibitory signal will then reduce autonomic nervous system outflow and subsequently aim to reduce blood pressure back down to normal levels. And here we can just see that we've got the firing of the baroreceptor nerve. You can see here that we've got these step changes to either 75 or 125 millimeters of mercury. You can see that you do have some dissipation or adaptation over time after this initial step change. So you have an increase in firing rate, and then it slows down here with 125 millimeters of mercury. You have a much more rapid increase in firing rate, and of course the dissipation is slightly less. But again, what we're seeing here is a dose-dependent increase in depolarization of membrane and subsequently having increases in firing rates ultimately, which are going to increase the amplitude of that inhibitory signal going to the brainstem. Now, when we look at the general pathways that these signals follow when they go to the brainstem, you can see here we've got the carotid sinus, we've got the aortic arch, two major baroreceptive sites, and those come in and synapse within the nucleus tractus solitarius, which we'll talk about in more detail in just a moment here, but largely in the brainstem or in the medulla. Here you have synapsing in the NTS, a bit of additional synapsing in other regions of the brainstem, which we'll discuss in a little bit more detail. And then ultimately with sympathetic outflow, having those go down the intermediolateral column, synapsing in the point of the paravertebral ganglia, and then having a long non-myelinated fiber going to the target tissues of either the heart, where we have beta-adrenergic receptor activation and increases in SA nodal firing rate, or we have them going to the vasculature to activate alpha-1 adrenergic receptors and subsequently increase vascular tone. When we look at these parallel signals that then go to the parasympathetic nervous system, they're, again, very relatively long preganglionic fiber here coming out of the spinal cord, and then having a relatively short postganglionic fiber here very close to target tissues. We think of, again, the vagus nerve or cranial nerve 10 being the primary one influencing cardiovascular function, and there the changes in parasympathetic outflow are going to largely influence heart rate through activation or lack of activation of muscarinic receptors, which are cholinergic receptors in the heart, and altering SA node and AV nodal firing rates. So broadly, this is the pathway that these signals are going to follow when you have changes in blood pressure. And so if we look at an example of baroreflex control of blood pressure, that's depicted here, where, for example, if we have an increase in arterial pressure, we'll have an increase in arterial baroreceptor activity. That increase in activity is going to result in an inhibitory signal going to the medulla and the cardiovascular nuclei within the brainstem. That increase in arterial pressure and the changes in firing rate in the inhibitory signal going to the brainstem is going to increase parasympathetic nervous system outflow. So what we'll have is an increase in vagal nerve activation, and that is going to result in a reduction in heart rate and a reduction in cardiac output. So again, trying to reduce flow to attenuate those increases in arterial pressure. And then also that is going to result in a reduction in sympathetic nervous system activity. That will result in a loss of the activation of beta-adrenergic receptors in the heart and be predominantly reduced stroke volume. And in parallel, have a release of vasoconstriction and reduction in vascular resistance, all of which are serving as a negative feedback loop to attenuate those increases in blood pressure that are sensed at either the carotid or aortic baroreceptors. Okay. So again, all of these signals are going to be passing through these peripheral pathways as we discussed last time, going through the preganglionic neurons, again, synapsing here, going to postganglionic and target tissues, and having modulation from visceral receptors coming back, and ultimately modulating these signals and optimizing them in the spinal cord. So now that we know that what's going on largely peripherally, we get the sensor, we get the activation of the baroreceptor or the loss of activation of the baroreceptor. That's altering an inhibitory signal to the brainstem. We've got a bit of a signaling cascade going on in the brainstem, but then much more detail going on with the peripheral autonomic nervous system and vasculature. So now that we know that's going on really outside of the brainstem, the question then is what goes on inside of the brainstem and how are these signals integrated effectively to alter sympathetic outflow and parasympathetic outflow. So the baroreceptors generally send afferent signals to the nucleus tractus solitarius, as we mentioned before. So that would be the NTS. And so within the NTS, you can see that we have a variety of signals here. So generally, we'll talk through these a bit here. But the NTS is really the integrator of these signals and sends, after these signals are received, sends signals to the rostral and caudal ventrolateral medulla. There, the RVLM sends out a pathoexcitatory signal. And importantly, the CVLM is not a primary driver of things, but it really is more of a modulator. So it takes in additional information from other sites in the brain, serves to modulate the ultimate outflow from the RVLM. And similarly, the caudal pressor area is also a modulator. So we have not just a singular reflex loop sending out either an increase or decrease in pressure. We've got inputs from a variety of centers within the brainstem and a variety of sensors throughout the body, trying to inform the RVLM what is the optimal amount or optimal change in sympathetic outflow or parasympathetic outflow that's ultimately going to be appropriate and return blood pressure back to a normal homeostatic level. So, of course, this is sort of a nuclei in a box depiction. This is probably a little bit more accurate, where we've got the NTS up here with parallel signals going on to the RVLM, CVLM being a modulator of the RVLM, certainly having other modulators come in here as well, baroreceptor and chemoreceptor afferents coming in, and those ultimately sending out signals to the body. A bit more physiological depiction here. Of course, the reality is that in an anatomically correct depiction, it's all sort of shades of gray here, but largely this is where these nuclei are going to lie within the brainstem, and the CVLM ultimately still provides somewhat of a break on the system and a modulatory impact on the system. So when we're looking at some of the other modulatory inputs that influence other parts of the brain and help to optimize output, or also in various scenarios can amplify these responses and contribute to pathophysiology, you can see here, again, we've got the NTS here with the RVLM, hypothalamus, spinal cord, and other areas here. You can see that in particular, as we discussed last time, so we've got the NTS. Interestingly, we have angiotensin 2. So angiotensin 2 is actually a modulator of quite a few of these pressor areas that can modulate pressor areas through changes in cytokines in particular, and changes in inflammation, as well as reactive oxygen species. And you can see that most of these things actually are pretty sensible, right? So here you've got cardiopulmonary mechanoreceptors. Fair enough, that's what we just discussed. We've got carbon dioxide, oxygen levels. So if you have hypoxemia, of course, that's going to be a pretty profound modulator of how much cardiac output and subsequently pressure you will need to optimize perfusion. Cytokines in the context of a disease, if you have elevations in inflammation, that generally amplifies a signaling cascade. Tissue metabolites, again, major influencer and modulator of many of these signals in the brainstem and autonomic nervous system outflow. And so collectively, again, sodium, cytokines, Wabane-like substances, cytokines, oxygen, and pH, all being significant modulators of outflow. And I would argue that, again, most of these are intuitive. So if you are, for example, retaining CO2, having reductions in oxygen, having very abnormally high levels of or low levels of sodium either direction, and having increases in inflammation, having excess metabolites, those are all things that are very intuitive amplifiers of sympathetic nervous system outflow. And again, angiotensin 2 being a major one here to have a central impact on influencing whether the body is going to try to maintain pressure and in response to a significant physiological or pathophysiological stress. Now, when we look a little bit deeper into modulatory feedback from the periphery, these group 3 and group 4 muscle afferents are major influencers of sympathetic nervous system outflow. And so these are chemosensitive and mechanosensitive afferents that are in skeletal muscle. So if you are out exercising very intensely, that's going to result in buildup of metabolites and stimulation of the group 3 afferents. Of course, muscular contraction will stimulate mechanosensitive afferents. Those are going to ultimately send a signal to the brain saying we're doing a tremendous amount of work here. We really need a lot more blood flow. And so therefore increased sympathetic nervous system outflow to cause constriction in regions of the body that are physiologically inactive, And then if that's not sufficient, then continue to try to increase perfusion pressure to those regions, largely by increasing systemic perfusion pressure and further increasing resistance in those inactive beds. So that's the role of those group 3 and 4 muscle afferents. And so there, we're really focusing predominantly on what are the afferent signals that are going to drive modulation of sympathetic or autonomic nervous system outflow. And it brings up a very important question, which is that is sympathetic nervous system outflow or autonomic nervous system outflow, is it all feedback and reflex, right? So it's just the baroreceptors and the chemoreceptors and everything saying, well, we've got dysfunction here, respond. Or is there also a feed-forward component to this? So, which would essentially say, well, when my brain is thinking about or taking in a significant sensory stimulus, or I know that I'm going to be exercising extremely intensely in a very short period of time, is there a priming feed-forward mechanism that is in place that ultimately serves to optimize the response to stress? And so here, the one of the best models here was really by Fred Eldridge, and so these studies were done in the 70s and 80s with the effect of motor cortex stimulation in the decorticate paralyzed and ventilated cat. And so the pivotal thing here is that Dr. Eldridge was able to find regions of the brain within the cat that have elicited fictive locomotion. So essentially a locomotor region of the brain, so the cat would walk even though the cat was decorticate. And the important thing now is that you say, well, now we can stimulate these regions of the brain that we know are there to elicit motor cortex movement and fictive locomotion. But when we paralyze the animal now, we have that stimulus to go exercise. The stimulus is say, I'm going to go do physical activity. But now there's no distal response, no muscular response at all. And we can get at the answer is, well, is there actually a significant feed-forward component here? And so certainly when we look at the cat, what you can see here is we've got under baseline conditions, we've got arterial pressure here. We've got the biceps femoris nerve for both the right and left sides. You can see that there's really, of course, no activity from the stimulation perspective. They've got integrated phrenic nerve activity here, and you can see here, so this is innervating the diaphragm. So the animal is ventilated and paralyzed. So this is just with the sort of endogenous level of phrenic nerve activity, and then end-tidal CO2, which is held as a constant here. And what you can see is that when you stimulate the brainstem in these regions that will elicit fictive locomotion, you can see that blood pressure goes up. So there's an arterial pressure response that occurs concomitant with stimulation of the motor cortex region. And again, this is just measuring the outflow to those muscular regions, not necessarily their physiological response. There's no physiological response because they're paralyzed. So you can see that you're getting this rhythmic signaling to those skeletal muscular beds consistent with a locomotion pattern. And then what you see here is integrated phrenic activity. So again, an apparent increased drive to breathe that's occurring concomitant with increases in the attempt to fictively locomote. And the entire time, the animal is being ventilated, so there are no changes in blood gases whatsoever. So this would really suggest that, at least in the cat, that when you think about exercising, when you have the central motor output to go and exercise, that in fact is sufficient to send a parallel signal from those motor regions to the NTS to ultimately increase blood pressure, so driving pressure, as well as preemptively increase phrenic nerve activity to say, well, I'm anticipating that I'm going to be having very intense exercise, so we should really increase autonomic nervous system outflow and the cardiovascular and respiratory parameters that will support that. Now, that's great for the cat, but then the question becomes, well, what about humans, right? And so when we look at muscle sympathetic nervous activity, so that can be measured in humans invasively using a microneurography. But here you can see that when we're looking at MSNA, you can see that we've got hand grip at either 15% of a maximum voluntary contraction or 30% of maximum voluntary contraction. Under normal conditions, you can see that 15%, we really don't get a huge muscle sympathetic nervous system response. But with 30%, we do. Okay, so this is you can see effective increase in force generation here with hand grip, and that's maintained this entire time. And you can see that towards the end of this, where you have metabolite buildup with 30% maximum voluntary hand grip, you start to get increases in muscle sympathetic nervous activity. So again, this is the skeletal muscle sending a chemosensitive, mechanical-sensitive signal to the brainstem saying, I'm getting underperfused here. You should start to redirect blood flow, even to a forearm. And but then in this study, what they did is they administered curare, and so curare is a paralytic, and so they administered that to the forearm. And what you can see here is that with muscle sympathetic nervous activity again, we have this increase, a small increase in force generation that subsequently dies off because this now becomes an attempted hand grip because the forearm is paralyzed. And what you can see is that compared to baseline conditions, you might have a little bit of an increase in sympathetic nervous system outflow. That's depicted over here, where you can see before curare, we get a very substantial increase in sympathetic nervous system outflow, which is dramatically attenuated with curare under both conditions here. So in humans, at least, it doesn't appear as though there's a very sizable sympathetic outflow component due to feed-forward mechanisms. However, when we look at the effect on heart rate, so again, I've got just for reference here, we've got mean arterial pressure here, but when we look at heart rate, you can see that now before curare, we get a very robust tachycardic response here, increase of roughly 15 beats per minute. And then with attempted hand grip, we have a very comparable increase in heart rate in response to the attempted hand grip, suggesting that there may be a feed-forward mechanism that is intact in humans that enables increases in heart rate despite the fact that there is actually no physiological feedback from the exercising forearm suggesting hypoperfusion. So again, very interesting observation, highlighting some differences between the human and the cat, as well as some mechanisms that might be conserved to increase overall cardiac output in response to intense feed forward and or intense mechanical effort in humans in the absence of that mechanical stimulus in the forearm. So again, when we go back to the brainstem now, we say, well, what actually goes on in the brainstem? So we've got the regulation of sympathetic outflow in various microenvironments. Of course, many studies have evaluated the role of nitric oxide or oxidative stress, and that's just depicted here, where when you have a good amount of nitric oxide that's generally protective, generally attenuates the effects of hypertension. Certainly oxidative stress is generally deleterious and augments some of these deleterious signaling cascades that result in hypertension. Now, some other specific reflexes that we haven't really touched on necessarily, we'll just sort of revisit in some in the context of volume depletion and volume loading here. You can see with the baroreceptors, when we look at, we'll just sort of walk through this. We've got volume depletion here, so you can see that as we deplete effective circulating volume here, we get a tachycardic response. Okay, and so that is largely baroreceptor driven. Of course, in the anesthetized dog, what you'll end up having is a also dependent dose dependent reduction in cardiac output. You have a significant reduction in stroke volume as well. So that reduction in stroke volume is due to losses in preload. And so collectively, you say, well, this makes some sense, that heart rate is increasing, stroke volume is decreasing quite dramatically. The change in cardiac output is somewhere between there because you've got this compensatory tachycardia. But when we volume load significantly, what you can see is that certainly in this preparation, that stroke volume is relatively well maintained. And interestingly, cardiac output tends to increase as well in a linear fashion, along with increases in heart rate. And that is due to the Bainbridge reflex, which is quite profound in canines. So here you can see that when we have atrial distension and an increase in atrial volume, that elicits tachycardia. And so that is a mechanism that enables the maintenance of operating central cardiac volumes. And functionally, you would say you don't want the heart to stretch sort of too much, if you will. And so that Bainbridge reflex, at least in this preparation, is quite profound and results in an increase in cardiac output, and largely and exclusively due to an increase in tachycardia. So hopefully that makes some sense in terms of how, certainly in the anesthetized open-chested dog, how these things can certainly be manifest in a graph such as this. Volume depletion being very straightforward, volume loading being this increase largely being reflex-driven in this preparation. So again, this is just, I think, a bit more detail. Hypovolemia resulting in a decrease in cardiac volume due to decreased stroke volume and despite a compensatory tachycardia. Hypervolemia, that increase in cardiac output, exclusively due to an increase in heart rate in the Bainbridge reflex due to plateauing of stroke volume. So hopefully this is giving you an idea, at least in some part, at least at a sort of basement level, how baroreflexes and reflex regulation of the circulation occurs and how that modulates sympathetic nervous system and parasympathetic nervous system outflow from the brainstem. And certainly look forward to discussing this more in our upcoming class periods. Thank you.